Well, hello, I'm Mark McClellan, Director of the Duke-Margolis Center for Health Policy. On behalf of Duke-Margolis, I'd like to welcome all of you to today's roundtable on advancing bacterial diagnostic development. We're really pleased to have you all with us for this important and timely conversation. Before we get started, I want to thank our staff who were involved in organizing and pulling together the work for this webinar, along with the distinguished speakers that you're going to hear from today. So thanks to John Bollinger, Marianne Hamilton Lopez, Nicholas Harrison for putting this together in collaboration with our speakers and other content contributors. I also want to thank Luke DeRocher, Ethan Chubb, and Derek Rapista as well. This webinar is part of our broader policy research portfolio, focusing on supporting, sustaining, and improving the market for novel antibiotics and bacterial diagnostics. And for this work, we want to thank the Wellcome Trust for providing support for today's event, and Schmidt Futures for more broadly supporting our ongoing policy efforts related to antimicrobial resistance and infectious diseases. So just a quick overview of today's program. Session 1 is going to focus on how diagnostic developers are generating evidence for regulatory review and some of the potential for improving those capabilities to get more confidence and as a result, more appropriate use of innovative diagnostics on such areas as harmonizing measures of clinical utility, for regulatory review, supporting healthcare provider utilization, aligning with current gaps in and challenges in payment models. Then in session 2, our panelists are going to discuss some of the challenges in the market for bacterial diagnostics, including general bacterial diagnostics, as well as those that identify drug-resistant infections and antibiotic susceptibility, and potential policy actions to address these challenges as well. So we're glad to have you with us. We want this to be interactive. The sessions are going to include opening comments, but plenty of time for a discussion. You'll hopefully see somewhere on your screen, probably at the bottom, the Zoom Q&A function. Please use that to submit questions or comments at any time. Like I said, we do have some time during our sessions for discussing some of these questions, and very often we'll hopefully have some respondents among our panelists who could contribute answers through Zoom as well. So please use the Q&A. To get us started, I want to provide us a few brief background remarks on the issues that we're going to be discussing in more detail throughout the roundtable. As probably just about everybody here knows, and that's partly why you're here, antimicrobial resistance is an increasingly important challenge in the United States and across the globe, and novel antibiotics, as well as diagnostics and surveillance systems, are all critical parts of the potential response to this growing challenge. And today, we're focusing particularly on barriers and opportunities related to the development of bacterial diagnostics. And many of you who join us today have worked with Duke-Margolis on some of the other issues as well, surveillance systems and support in public health and healthcare organizations for better surveillance, incentives, especially pull incentives around new, potentially highly valuable antimicrobials. But today we're focusing on the development and adoption of innovative bacterial diagnostics as part of this comprehensive response. This slide shows from a recent Lancet report the growing global burden of bacterial antimicrobial resistance. The report split that burden into deaths attributable to antimicrobial resistance, deaths that would have been avoided if effective antibiotics were available for the resistant organisms, and then also deaths associated with, not attributable to AMR. That is, deaths that could be avoided if those infections were preventive, which is something that surveillance and diagnostics could potentially help with as well. As you can see, a lot of burden here, 1.27 million deaths attributable to bacterial AMR, nearly 5 million associated with bacterial AMR in 2019 alone. In the United States, close to 50,000 attributable deaths and close to 200,000 deaths associated with bacterial AMR, all illustrating the significance of the AMR challenges and building on a lot of previous estimates that have this that portray the same basic picture. Now, on the next slide, we've seen these impacts getting worse over the last 3 years in conjunction with the COVID pandemic. We've actually been seeing a decline in AMR-related deaths over the 5-year period prior to the pandemic in the U.S., a modest one, reflecting such steps as some progress on antibiotics and effective systems for managing antibiotic use, et cetera. But during the pandemic, the CDC identified some very concerning increases in healthcare-associated infections resistant to antimicrobials. So this is a worsening problem. It's been made worse by the pandemic. And it's just a reminder that we need effective infection prevention and stewardship, which in turn would benefit from more effective and accessible bacterial diagnostics. So let me just turn more specifically in the next slide to the role of diagnostics in combating antimicrobial resistance. These diagnostics can help address the rise and the spread of AMR by better informing antibiotic prescribing. That'll help us preserve the effectiveness of current and potential future antibiotics. And diagnostics can also help with bolstering effective surveillance efforts. This helps helps prescribers make the most appropriate antibiotic decisions, reducing the use of ineffective or unnecessary antibiotic therapy. The diagnostics can help improve individual patient care through that mechanism. And this also helps mitigate the pressures that drive the development and propagation of antibiotic resistance. And then diagnostics can also help us identify concerning infections, progress or developments in resistance mechanisms, and then take more effective and informed steps to prevent and contain their spread in particular communities and hospitals and settings within them. And finally, accurate and available diagnostics underpin surveillance efforts that enable us to measure incidence and trends related to antimicrobial resistance. So not surprisingly, in the next slide, diagnostics is recognized, diagnostic development is recognized as a critical part of the United States National Action Plan on antimicrobial resistance. As the diagnostics pillar here highlights, diagnostic supported stewardship helps ensure that patients get the right drug, the right time, the right route, et cetera. Diagnostics help improve the surveillance efforts that I just mentioned. And the National Action Plan acknowledges the importance of prioritizing the development and use of rapid, effective diagnostics. And in fact, on the Action Plan Goal 3, in the next slide, specifically focus on the role of rapid, effective diagnostics, more effective diagnostics, which can speed appropriate care in the inpatient setting, can improve antibiotic stewardship in the outpatient setting. Innovative diagnostics can combine bacterial identification with susceptibility in a single test, potentially much more quickly, test for pathogens in a rapid multiplex fashion, or offer the ease of a direct from sample diagnosis. So that's the potential. We've seen how improvements in diagnostics have had a huge impact on COVID and other major infectious disease threats, and these new diagnostic technologies also have implications for enhancing our ability to combat antimicrobial resistance. Of course, innovation here has a high bar, especially for serious infections. You want a test that is highly sensitive to make sure that serious resistant organisms aren't missed, and ideally highly specific to advance that goal of effective antibiotic stewardship. So this is not easy. And there are a number of barriers on the next slide to advancing bacterial diagnostics. Today's webinar, we're going to talk about some of these, just a few quick thoughts up front. First, and I want to thank IDSA for a very helpful recent report on the role of diagnostic tests that is a nice job for presenting many of these issues. Number one, we have an evidence problem, important gaps in available evidence on how available diagnostics can best improve patient care, improve public health, can drive those improvements in decision making for individual patients and overall surveillance and policies related to antibiotic use. It is not necessarily straightforward to develop this needed evidence, especially in real world contexts with real sick patients and very sick patients in hospitals, where the timely diagnostic and accurate information is really critical. Adding to these evidence limitations, there are limited resources to support clinicians in diagnostic stewardship and to help guide clinicians in decision-making based on results of these increasingly advanced and sophisticated diagnostic tests. And as with novel antibiotics, our current mechanism for paying for diagnostics on a fee-for-service basis provides only limited return on investment for the diagnostic developers. And just as people have, I think, widely recognized now in the context of therapeutics, the payments for diagnostics also don't reflect the broader value when you use a diagnostic effectively, not only for that patient who's being treated, but for the impact on stewardship, the impact on population health emerging from better containment of an infectious disease, of a serious infectious disease threat. And so that leads to some potential policy solutions, which I'm mainly going to leave to our upcoming panel. There's a nice slide summarizing it, steps to streamline and update regulatory pathways through, for example, better incorporating the improving capabilities in real-world post-market evidence, updated breakpoints for drug combinations, FDA, CLSI alignment along with that. Public-private partnerships to develop better registry systems, clinical trial networks to support that evidence development that's so important, and then reforms in reimbursement to address the reimbursement challenges that I described earlier, both pull incentives such as tax incentives for priority innovative technologies like we're seeing in other areas around advanced biologic manufacturing platforms, et cetera, and some therapeutics, and both push technologies, I should say, to help advance them along, and pull technologies, reimbursement tied more to the value, the impact of these tests, not just their cost of production. So we'll hopefully get into a lot of these issues today. And with that, I'd like to turn this over to, I think on the next slide, to Marianne Hamilton Lopez, who will be managing our first session today on developing bacterial diagnostics that combat AMR. So Marianne, over to you.
All right, thank you so much, Mark. And good afternoon, everyone. I would also like to thank you for joining us. Once again, I'm Marianne Hamilton Lopez. I'm the Senior Research Director at the Duke-Margolis Center, and I'll be moderating our first session on developing bacterial diagnostics that combat AMR, regulatory review, and clinical utility. We're joined by a wonderful group of speakers, and I'd like to begin by introducing them. So today we're joined by Vance Fowler, who's the Florence McAllister Distinguished Professor of Medicine at Duke University School of Medicine, Robin Patel, Elizabeth P. and Robert E. Allen Professor of Individualized Medicine at the Mayo Clinic, Melody Domerad, Vice President, Medical Affairs at Day Zero Diagnostics, and finally, Fred Tenover, Vice President, Scientific Affairs at Cepheid. So we're going to ask that each of our speakers start with some opening comments. Next, I'll turn it over for opening comments from Vance.
Thanks, Marianne. Could I have the first slide, please? perfect. While he's getting that put in slideshow mode, I appreciate the opportunity to speak today. And my task is to sort of put the context of the clinical context of how diagnostics and AMR interact. I'm not able to see the slides. I hope everyone else can. Okay. So I think the key thing to remember with this is that, clinicians ultimately use antibiotics for a variety of reasons. I can assure you that selecting AMR is typically not highest on their list of reasons for using antibiotics. And until we are able to provide diagnostic information that can help clinicians safely withhold antibiotic use, we're going to be challenged with this issue of AMR. So those are my disclosures. Next slide. Okay, so the first point to underscore from the clinical perspective is that we treat syndromes, not bacteria. When the decision to start someone on antibiotics is made, you don't know what you're treating. You have this. You have a person who's been in the ICU for sometimes weeks, who suddenly develops a fever, their oxygen requirement goes up, and you have this chest X-ray. That's what you have, and you have to make a decision. You don't know you don't know that person has an ESBL, Enterobacteriaceae, or an MRSA, pneumonia, or what have you. You know that they're acutely ill, and it's likely to be a bacterial infection. So the immediate pressure is to make a decision on antibiotics and start treating. Next slide. And so there's more clear data showing that making the initial choice wisely impacts outcome. So these are data from a long time ago, but they clearly underscore in the green bars shown in the figure that patients who receive inadequate initial antibiotic therapy are significantly more likely to die in the context of critical illness than those who do not, those whose initial antibiotic therapy is effective, shown in the blue. So if you're going to make a choice, you need to make a good choice first. Next slide. And time also matters. So this is interesting data demonstrating an association between timing of initial effective antibiotic therapy and survival in critically ill patients with septic shock. So as you see on the x-axis, being hours, survival drops, which is shown in orange, drops significantly over just a few hours from zero time on the left up to just 4 or 5 hours in the middle, and that the timing the time to effective antibiotics was directly tied to better survival likelihood for critically ill patients. So there's an emphasis to treat, there's an emphasis to choose wisely, and there's an emphasis to choose quickly. Next slide. And as a result of that, the current guidelines for syndromes such as ventilator-associated pneumonia, such as we've talked about, are to essentially emphasize the fact that you get points for being right and not for style. So these are the suggested empiric guidelines, which recommend treating with not one, not two, but 3 different antibiotics while you're trying to ultimately find out what you're treating. So, so the emphasis from a clinical standpoint is to treat. Okay, and next slide. And there's even more emphasis, which is not raised, but as frequently as some of these other points, but is an unfortunate reality, and that is that folks are watching. So that increasingly, if there's a bad outcome, there are the issue of medical legal risk is essentially always hanging over physicians making decisions with incomplete data. And I literally get this as a redacted approach from just a week or so ago. And, you know, I probably get a couple of these a month, and I suspect many of the clinicians in this audience do as well. So next slide. And so I'd summarize my section for antibiotic rules of engagement is the following. The initial treatment is a guess. You don't know what you're treating. And if you're wrong, as the saying goes, you're dead wrong. So the emphasis is to try to maximize the chances that from an antibiotic standpoint, that you're providing at least one drug for which the organism has susceptibility. That leads to antibiotic overuse because you get points for being right and not for style. Antibiotics also treat fear as well as infection. You're treating syndromes, you're treating the unknown. And that clearly contributes to the growing problem of antibacterial resistance and overuse. Physicians will, and to my estimation, should continue to prioritize the patient in front of them rather than this simultaneous conflicting pressure and emphasis of the greater societal impact of those antibiotic choices. And ultimately, the critical data is, as we talk about these diagnostics things over the course of this meeting, is I need to know how to kill the pathogen. I don't need to know its name. And in fact, things like knowing that it's a Staphylococcus aureus, or knowing that it's an E. coli, or knowing that it's a Pseudomonas, are essentially surrogates for providing clues of what are the best choices to treat that bacteria. And as a consequence of all this, our best option in 2022 is typically to start broad and de-escalate as the data comes back from our current conventional diagnostics. And I'm suggesting that in the meeting today, that diagnostics provide a clinical possibility of improving diagnostics in AMR by confusing the issue with facts. And I appreciate the opportunity and look forward to the discussion. Thank you.
Thank you very much, Vance. I'll turn this over now to Robin.
Thank you, Marianne. I love diagnostics. This is what I think about and do all day long. And we're in a time when there's really a rapid revolution in new diagnostics. We have to figure out what we need to serve Vance and his colleagues and our patients. And then we have to figure out how to use them. And it's complicated. We're dealing, as Mark mentioned, with many kinds of organisms and many antibiotics and many kinds of resistances and many kinds of syndromes. So sometimes we have to carry out research to answer questions. And what I wanted to take just a few minutes to do is to highlight some of the work that the Antibacterial Resistance Leadership Group, of which Vance is leading, and I'm the Laboratory Center Director, has been doing in the area of diagnostics. So I wanted to share with you 7 of our many studies that we're executing or have executed in the area of diagnostics, just to sort of throw out some ideas about where we maybe need to go in this area. The first has to do with assessing the value of diagnostics. We are being delivered some fantastic diagnostics from diagnostics companies, but sometimes we don't know if we should use them or not and how we should use them. And so I'm going to show you 3 studies, 2 of which we've already executed that are randomized controlled trials. That's what we do when we have a new therapeutic. We do a randomized controlled trial, but the idea of doing randomized controlled trials with diagnostics is somewhat new. Although this first study we did was with one of the first multiplex PCR panels for testing positive blood culture bottles, where we randomized patients who had positive blood culture bottles to have this test or not. And then we had a third arm with antimicrobial stewardship plus the rapid test. It was really innovative and paved the way, I think, for figuring out how to use this kind of testing in clinical practice that now, based on some of our recent survey data, is pretty common. I'm not going to show you the results. I don't have enough time. We also did another study, next slide, please, where we did a similar study of patients with gram-negative bacteremia, but here with rapid phenotypic susceptibility testing. And later in the discussion, we can talk about phenotypic versus genotypic in gram-negative bacilli. Phenotypic susceptibility testing was, I think, a lot more meaningful, at least at the time. And again, we can talk about that. So we did this study where we compared a novel platform that uses real-time imaging of bacteria to generate phenotypic susceptibility results called the Accelerate Pheno, compared to control, looking at patients with gram-negative bacteremia, positive blood cultures, that is, and we showed a benefit in terms of antibiotic utilization in the Accelerate Pheno arm. Again, I don't have time to show you all the data. And then currently, next slide, please, we're executing a follow-on study of that called FAST Antimicrobial Susceptibility Testing. It's a multi-center prospective randomized clinical trial with another rapid phenotypic susceptibility platform called the Reveal from bioMérieux. And this will be carried out outside of the United States at sites with fairly high rates of resistance in gram-negative bacilli. So I wanted to share with you this randomized clinical trial design. Next slide, please. Another way of looking at new diagnostics, and Melody spoke to this earlier, is platform studies. And so I executed a platform study. I didn't build a slide around it for extragenital Neisseria gonorrhoeae and Chlamydia trachomatis that allowed FDA clearance of the first tests with this indication. And currently we're designing and going to execute a master protocol study for evaluating diagnostics that go directly from blood for diagnosis of bacterial infection. And again, I don't have time to talk in detail about this, but I do think that master protocol designs can be very helpful in terms of bringing companies together to generate the data in a single clinical trial to get the data needed for approval. I'm happy to talk more about that later on. Next slide, please. We're also really interested in host response diagnostics. You know, in clinical medicine, and Vance can attest to this, we have erythrocyte sedimentation rate and C-reactive protein and procalcitonin. But, you know, today with transcriptomics and proteomics and so forth, we can do so much more. And so we're looking at a platform that looks at host gene expression to diagnose acute respiratory illnesses as being bacterial or not, because we know that there is a lot of antibiotic use to treat viral infections, which makes no sense. But sometimes it's hard for clinicians to know that it's not a bacterial infection. So perhaps interrogating the host response will be the answer to that. Next slide, please. And then we're generating a lot of sequence data in our ARLG studies. And we think that once we better understand genomic sequence data, we can use that data to guide Vance and his colleagues in terms of managing his patients. In this platform called Genosmart, it's a platform for taking whole genome sequence data of bacteria and comparing that whole genome sequence data to whole genome sequence data that we have from our ARLG studies. And the information output is, of course, you know, what species is it, what antibiotic resistance genes are present, but also what's the phenotypic characteristic of an isolate with that kind of sequence, and what are the clinical outcomes of patients who are infected with related organisms. So this is earlier on than an interventional study, but we think that this is an important area to expand our knowledge in and to provide a service really for analysis of whole genome sequence data. And finally, last slide, please. We're aware that with a lot of the novel diagnostics that go out there, some of the reports are fairly complex. And you heard from Vance early on about what his needs are, but we're aware that sometimes there's so much information coming up from the lab that clinicians just don't know what to do with it. And a great example of that is reporting from positive blood cultures. I mentioned earlier that on the basis of some of our studies and other people's studies, there are a lot of rapid diagnostics that are used on positive blood cultures. But we were really interested in figuring out how people are are issuing those results from the lab. And so we have a study that's currently under review that was a survey of labs across the United States and some outside of the United States in terms of how they're reporting. And there's a great diversity of ways labs are reporting, some of which are not helpful. And so we ended up with some best practice recommendations on when to use this kind of testing and how results should be reported, and also what laboratories should do with discrepancies and so forth. So I think some of it's on us in the laboratory as well to figure out what to do with new diagnostics. And I think I could talk all day long, but I will end there. Thank you, Marianne.
Thank you, Robin. All right. And then finally, I'd like to turn this over to Fred for his opening comments.
Great. Thank you so much, Marianne. And if I can share my screen. Today, what I'd like to do is basically give you a sense about the complexity of developing tests for resistance, and then also why we really need partners in order to do this well. Hopefully everybody can see my screen here. Trying to get it into, there we go. Okay, so let's start out with the idea that you have this novel antimicrobial resistance test and you want to get it into the FDA's hands for clearance. But the fact is, it's becoming increasingly difficult to do this. Now, why is that? Well, there's several things to consider. The first thing is just the complexity of resistance mechanisms that we're seeing now for each antimicrobial class. Onto that, we layer the number of sample types, the different transport media that may be used. Do you want this to be CLIA waived or not CLIA waived? And then lastly, accessing the number of specimens you need, because the number of laboratories willing to participate in clinical trials is actually going down. COVID really decimated a lot of our clinical trials, and we need to understand better how to improve this. So the question is, should companies undertake this whole registration process by themselves, or should they be working with partners? And if so, what partners are available? So today, briefly, I just want to talk a little bit about using partners to generate the data for your FDA submissions, and then how to potentially improve or facilitate that whole regulatory process. So I wanted to start just with a sense of what we have out there now. We have some surveillance tests, and I'm showing you just the carbapenem resistance genes in those. We have several blood culture panels. Again, the carbapenemases that can be detected by these, and we have some pneumonia panels. Now, what I want to emphasize to you is when you look at 5 targets, you're not just talking about 5 primer pairs and a probe. And I think to help you understand this a little bit better and the complexity, I'll talk just a little bit about one product out there for carbapenem resistance. This is the Expert CARB-R test. It detects 5 families of carbapenemase genes, the KPC, NDM, VIM, OXA48, and IMP. It has 3 sample types that are cleared, rectal swabs and perirectal swabs, as well as testing colonies that have already been shown to be carbapenem non-susceptible. So this test has an infection control side. It also has an antimicrobial stewardship side. But to get all of those things on label basically took about a year to complete the regulatory process. So here's just another way of looking at it. And I want to emphasize again that the starting point for a lot of resistance tests is 50 positives for every target for every specimen type. That's rather daunting, but in fact, that was the starting point for this particular test. The reference method was culture plus sequencing. So every organism had to be sequenced, and any specimen we used, like a rectal swab that was negative, we still had to carry out 5 sequencing reactions on that to prove, in fact, that there was nothing hiding there. And on the right-hand side of this slide, I'm just showing you a partial list of all the organisms we had to pull together in order to do contrived specimens, because there was no way we were able to find throughout all our clinical trials 50 IMPs, 50 VIMs. KPCs we could get, OXA48, we came close, but everything else required contrived specimens. And the other thing is we're not just really talking about 5 genes. We're talking about genes with a lot of alleles, a lot of variants. And you need to know that your product is detecting not just KPC1, but all the KPCs. Now, how much of a challenge is that? Let me just give you a sense. Back in 2001, my team described KPC1, the Klebsiella pneumonia carbapenemase. That was an organism and a resistance gene that actually came out of a freezer from 1996. But where are we now? There's 126 variants of KPC. So that is our challenge now when we're talking about developing tests for resistance for the carbapenemases. There's a lot of them there, and some of these are quite variable. So what can help? A government partner, the CDC, FDA, Antimicrobial Resistance Bank. This is a really helpful tool. It has a lot of emerging antimicrobial resistance genes in a variety of different bacterial species. They've all been confirmed by DNA sequence analysis, and they're continually adding to this collection. So now, what if we want to take this test and we want to make it even broader? This is where we really need academic partnerships, because all of the variants that we want, all of the genes we want to see, the ones we want to detect, they are not all in the CDC FDA data bank. There are a large number of emerging resistance genes, and pretty much the only way to get to those is with academic partnerships. So this was a study we did early on with David Nicolau and his team at Hartford Hospital. It really helped us to understand if we were on the right track, especially looking at the emergence of carbapenemase producing Pseudomonas aeruginosa isolates. So now we have the data, now we have to get it through the FDA. And this is where something novel, the public-private partnerships using the Independent Test Assessment Program, which is in the NIH. So this is a partnership that theoretically, I will say, could help us shepherd submissions through the FDA. Their mission, and they started actually just in 2020 to the best of my knowledge, is to accelerate the regulatory review of products specifically for over-the-counter testing for COVID. And they were very successful in moving products on a number of new platforms, some of which are listed there. If you go to their website, it really helps you understand, I think, the flavor of what they're trying to achieve. And this is all through their Rapid Acceleration of Diagnostics work network. So you have experts from government and academia and industry. They can help you really assess the best way to do studies and get those studies done. Their focus, however, right now is limited to over-the-counter tests, but it's the whole ball of wax from data collection to data management to FDA submission. They help with this pathway, taking something all the way through in collaboration with the FDA every step of the way. So the question I think we need to explore is, can something like ITEP and RADx be utilized in the future for antimicrobial resistance tests in order to make sure that we get them through in a timely fashion and we get these important tests, as Dr. Fowler has pointed out, into the hands of clinicians so that we change empiric therapy to definitive therapy as soon as possible. So going forward, I think we're going to need partnerships, and we're going to need partners who can just not help us generate data, but get that actually all the way through registration with the FDA. So let me stop there. Thank you.
Excellent. Thank you so much, Fred. And if I could just have the rest of the panelists come back on screen. Perfect. We still don't see Melody, but hopefully you can join us. There she is. All right. Thank you so much. So I think this was an excellent introduction to a number of the issues that are faced, especially by developers, as they are looking ahead at the regulatory process. The goal of our session here is to really think through, are there some strategies that could potentially be used to make this a more efficient, streamlined, process for those who are thinking about the development. And then as Vance put forth in his comments, those who are going to have to actually implement these types of diagnostics in their everyday care. So I wanted to start, Fred, from a comment that you made towards the end of your presentation, where you kind of said, and now we have the data, and then you could move on. But to get to the now we have the data, you explained a number of different banks that you had to go through, as well as developing and implementing these academic partnerships, because there's data at different locations and it's not easily accessible. You know, how does that process kind of fit into the overall efficiency of the development of these diagnostics? And is there a way that we could ask for more transparency or more streamlined for those who are trying to develop diagnostics currently?
Sure. Thank you for the question. And also kudos to the ARLG for the terrific work that they're doing in moving diagnostics forward. You know, I think I mentioned academic partnerships because one of the things that developers of diagnostics are trying to do is fill an unmet medical need. And so early on, we want to work with clinicians to say, this is what we're thinking. Is that what you really need? And getting that initial buy-in, I think, is very, very important to make sure that we're on the right track, because we want these tests to actually improve the delivery of patient care. And we do have a lot of challenges in knowing what the best tests are. What markers do clinicians really need? And are they going to be effective at predicting susceptibility or resistance? We know that the presence of resistance gene is normally, although not 100% correlated with phenotypic resistance. But the question is, how do we view a negative gene result? Can we presume that that organism is going to be susceptible or not? And I think if we're very savvy about pairing resistance genes and organisms and antibiotics within the context of a given medical center, we can make those negative predictive values meaningful. So let me stop there and allow others to comment.
Yes, Robin.
I can comment, and it's really just jumping off what Vance had said and some of what I shared as well. I think that clinicians need information that really tells them what they can safely and effectively do, rather than just giving them a lot of scientific information about what genes or mutations or names of things are there. And I really liked the way Vance pointed that out. And it kind of aligns with what Fred is saying as well. You know, that's a big ask, because you have to take what has been detected and kind of translate it into, okay, this means you should do this. And that's what I was after when I was talking about that last study where we were looking at reporting of results from multiplex panels, which is all over the place in laboratories across the US. And so now we have an opportunity at least to try to give out some best practice guidelines in that area. But I will also say that sometimes we don't know what we need. And I'm going to speak, I'm also an infectious disease clinician. You know, if you ask 10 infectious disease clinicians what they need, you probably get different answers. And honestly, Fred, if you asked a lot of people like back in the 90s if they needed this, right, you might have gotten some interesting answers about that. If you ask them today, I'm sure, or their kids, I'm sure you'll get a different answer.
So I would agree with everything that has been said, but I think that there are additional considerations, and that clinical trials are expensive and challenging for all of the reasons people have said. Building into the clinical trials longer-term follow-up. So clearly you want the data that can help you get to the FDA, but much of what physicians want to see are not just, okay, what did it mean when we identified that pathogen and which drug it was best treated with, but also did it affect the length of stay in the hospital? And what about the insurers who want to know about return of the infections or longer term issues? A clinical trial that can be designed to have an analysis that allows you to give the data to the FDA to say, yes, we can identify, we can find that drug, but also the longer term concerns that help physicians decide, yes, that was really useful. The X number of patients had a half the length of hospital stay as others. that a clinical trial and all of our concerns should be trying to answer multiple questions.
That's a great point, Melanie. It's kind of the ideal of what we would hope, that the diagnostics could be part of our long-term surveillance activities. And how do we ask so much of our clinical trials, and our developers, and are they thinking in that direction? And if not, how do we get that broader vision of saying we're not just getting this through regulation? That's not the end of the story. It's going to be also, is it going to be useful for advance? And so do you feel like that's already where we are, or do we have some more work to do?
I think there's always more work to be done. And I agree with Fred that things like banks are critically important. They don't necessarily have the long-term follow-up that we need. So I don't think there is a single solution. I do agree it's getting more challenging to get hospitals to have longer term studies, or just to be involved in general in this prospective collection. I think it's a worthy effort, though. Clinical trials are expensive in terms of money, time, effort, and maximizing the use. But is there work to be done? Absolutely.
Anyone else want before we move on? Well, just a question about kind of the private public partnerships that were already discussed. So I think my question, maybe perhaps for Vance, is, you've talked a lot about, after we get through the regulatory process and then trying to implement often these novel diagnostics. What's a better process for doing that? And can that be, we heard some examples from Robin about what is already kind of ongoing studies, but where does the clinician fit into those?
Well, I think ultimately the clinician is the, I don't want to say the sort of terminal user, but they're going to vote with their feet in terms of the utility of the diagnostic in question. So I think the clinical questions to be posed kind of organically arise from the unmet need. Either you do something, you give an answer that wasn't possible before, or you do it more accurately, or you do it more quickly, or I won't use it, because why should I? How are some sort of public-private partnerships that kind of address that? I know Robin had given a list of several really nice examples. There's some others that actually led to FDA indications, FDA approval. So one is the specific of which is being Mastermind GC, where we looked at the syndrome of extragenital gonorrhea and chlamydia trachomatis. Neisseria gonorrhea is one of the identified pathogens of drug resistance and particular problem. If it's outside of the urogenital tract, it's a real tricky thing to diagnose. And in a long story short, in 2014, Fred and I had a presentation at a meeting not that different from this. FDA, NIH. I had kind of noticed in our lab, I'd heard from the labs, the diagnostic companies, it's really expensive to do these trials. And then I noticed in my own research lab that I would use these samples from one patient of a particular syndrome or what have you. I'd use those biological samples sometimes multiple times to answer multiple different questions. And so this obvious sort of question, largely probably of ignorance on my part, just because I, was like, well, why in the world can't we have one patient inform multiple diagnostic platforms? And I proposed it, and I honestly didn't know that much about extragenital gonorrhea, to be perfectly honest. It's not my area of research. But Dr. Tinover tracked me down after and said, this is where you need to try it. And long story short, we undertook the trial. We enrolled 2,500 patients and ended up with not one, but two different FDA approvals from that from that experience, so that the patients were doing just that. You know, Melody mentioned a lot of those details that you gotta iron out. What order are they going to be? The samples going to be obtained, and there are ways around it. And it's also syndrome specific. So the issues that arise from bacteremia, you know, Dr. Patel is talking about some of those that we're designing now. They're a lot different than, you know, than urine or extragenital GC or just what have you. So they're going to be syndrome specific issues. But I think that might be one means that can help with advancing some of this beautiful technology that's out there. And something where along the lines is, okay, the federal government, whether it's through NIH, whether it's through AHRQ, whether it's through CDC, what have you. Obviously, I'd like to see it come through ARLG, but you know, but that some entity would take on a trial and said, okay, we're gonna conduct this trial in, you know, insert your syndrome of interest here, hospital-acquired ventilator-associated bacterial pneumonia, and that the companies, you know, can partner with us. We can fund the enrollment of the studies. They have to play nicely one with the other. There's, you know, issues of IP and all sorts of complicated things that you can get around, but that, you know, they would provide the necessary sort of requisite supporting information for their, you know, their respective platform. We work out a means by which to, you know, all parties can agree. And then you enroll the trial and do just what we did with extragenital GC and other syndromes. And you know, the advantage from the government side is that you can stimulate meaningfully advances in diagnostics by, you know, making these platforms available. And there's the advantage from the industry side of having access to a trial that might not otherwise get funded. I'll stop there.
What do you think? Did Vance solve it?
It's on the right track.
All right.
You know, I think, yes, the mastermind study, we participated in that, and that was terrific because it really did serve an unmet medical need. And that is looking at, you know, gonorrhea and chlamydia outside the genital tract, looking at throat and rectal. I think that we have a lot of unmet medical needs now, and there's more of them because we're dealing with resistant pathogens. One might ask the question now, well, should the next follow-on study be molecular methods to identify resistant gonorrhea strains, so that if it's susceptible to fluoroquinolones, we can give a single oral dose of Cipro and be done versus giving somebody a shot of ceftriaxone. But again, you know, when you're looking at this, you identify the strain of gonorrhea, or you identify gonorrhea being present in a clinical specimen. Now it's a subset of that you're going to ask the question and say, okay, is that now a resistant strain? And so far, it's been really difficult to make the case on a return on investment to do that as a diagnostic manufacturer. And yet, in my mind, it's an unmet medical need because of the rising resistance rates in gonorrhea. So I think being very strategic going forward about what resistance mechanisms, what resistance detection methods we need for which clinical specimens or syndromes is something that we need to think carefully about. Just think about where we use antibiotics poorly. Urinary tract infections is one. Do we need some way of identifying resistant organisms directly in urine in less than 30 minutes in an emergency department in order to optimize therapy for that group? These are the sorts of questions I think we need to be answering. And it could be that this is not one company, but multiple companies that can weigh in here, maybe under something like the ARLG format. But we need to move beyond now just detecting organisms to pairing those with the appropriate resistance genes so that we can tell clinicians very quickly, this is the way to go, or perhaps, hey, you know what, antibiotics are not needed here.
Yeah, Robin, go ahead.
Yeah, I just want to follow on with something that Fred said that I think is really interesting. And I'll poke a little bit at Mark, because I think on one of his slides, he said diagnostics to detect resistance. And I think that what Fred is pointing out is that diagnostics to detect susceptibility, where we can use some of our older antibiotics with comfort, and probably with a longer track record of safety, right, is and probably improve cost, a lot of beneficial areas is really something to explore. But I also hear what Fred is saying is that, well, we've moved on, so clinical practice isn't using those drugs anymore. So there's not maybe that much interest and impetus in going in that direction. But I really think that's a great approach, is that, you know, a lot of people don't have drug-resistant infections, but everybody's sort of being managed as if they do, for the reasons that Vance nicely outlined at the beginning, at least initially. But if we could begin correctly, we could repurpose some of our older drugs.
And Robin, while I have you, kind of going back to your opening comments, and you were talking about the use of the master protocol as a tool, and now you thought there was a lot of potential there, and how do we or the master protocol, what were the things you wanted to say when you thought you didn't have the time, but how does that become more of an integral part to make this a more efficient process for development?
Well, I think that Vance really nicely outlined what we did with Neisseria gonorrhea and chlamydia trachomatis for extragenital infections, throat and rectal diagnostics. And there were no diagnostics that were FDA cleared for those indications prior to the study that we did. As I mentioned, we're now doing a mastermind study for direct from blood diagnostics. Those are in their infancy, and quite frankly, it's a lot harder of a study than the original study that we did with Neisseria gonorrhea and chlamydia trachomatis. But I think the idea of doing this for many infectious diseases makes a lot of sense, right? You collect well-characterized specimens from a cohort of patients and you divide them up, or in the case of swabs, we collected multiple swabs. In the case of blood, you collect enough blood to be able to divide up and so forth. And then many companies can work together to generate the data they need to get clearance approval through the FDA all at the same time, working together. To me, it's like a lot more efficient from a financial standpoint overall, if you think about it. And I think there are a lot of places where this could be used, and it would, save a lot of trouble for companies kind of redoing the same thing and maybe competing to perform their studies at some of the same sites as well.
Well, I'd like to hear responses from those who would be asked to work in multi-company kind of collaborations. Melody and Fred, how would this be implemented?
Well, we welcome it. So Cepheid was one of the partners in the mastermind study, and we did get FDA clearance. And we frankly couldn't have afforded to do that on our own. We sort of took the bent that, golly, a lot of laboratories that want to do this, they're validating it on their own, but it wasn't enough. I mean, having spent almost 20 years at CDC, you can never take your public health hat off once you've been in public health. And it really was, I think, an unmet medical need. And so having the ability to go through the ARLG to get this done, I think was helpful. And I can think of a lot of other studies where similar things may be of real value from the diagnostics, of bringing multiple manufacturers to do head-to-head studies with the idea that we want to get them all on label. This is not ours is better than yours. It's we all have comparable products, but we need better coordination for clinical studies. So I think we should be doing more of this, I think, to promote diagnostics broadly.
I would agree. And I think the competition for sites and the competition for time is not an insignificant factor. And rather than having multiple companies or other research groups, not necessarily industry, all seeking, there are certain things that you need. If you're working within the emergency department, it has to be a hospital in which you actually have a study coordinator who is there. You don't have the time, if you're trying to get the sample before antibiotics are administered, to buzz someone from another building or the third floor for that matter. So having someone and having a well-coordinated laboratory, if you are collecting blood samples for 5 different protocols, one needs to sit on the clot, great, but 2 of them need to be spun immediately. One has to be put into -80. You need an octopus of a study coordinator. They're not an endless variety of sites who can handle the complexities of what amounts to a multiplexed protocol. So doing it as a collaborative, it simply makes the best possible sense for everybody.
There's one other potential advantage to be gained from working together in these master protocols, and that is particularly in syndromes in which you don't know what you're treating and you don't know what you're diagnosing, the prospect of developing a reference standard, a gold standard. And Robin may want to comment on that because she just submitted a beautiful paper on that really complicated issue. But if you could basically take, a couple of different diagnostic companies, you have 3 diagnostic companies, you have any 2, if you get 2 agreeing results, then that becomes your reference standard, for example. But Robin, you want to speak further on that?
Absolutely. I think when we were diagnosing extragenital Neisseria gonorrhea chlamydia trachomatis, it was pretty easy. We were after 2 organisms, but we could look at the results of the other assays and compare them to the one that we were generating data from. So we could gain some additional information about what the truth is. If you have a positive result, but maybe whatever you're using as your reference standard, say it was culture or something like that, which isn't great, isn't comparing well, how do these other diagnostics compare? But that's, relatively simple. As we get into some other infectious diseases like pneumonia, for example, or urinary tract infection even, the current standards that we use to diagnose those infections are very poor. And so when you bring on what is purported to be a better diagnostic and then try to compare it to standard of care, knowing that standard of care is not very good, that's going to potentially make that new diagnostic look poorly sensitive or poorly specific, depending on the scenario. I can spend time going through those, but I think you follow that. And so once you have more data and you're putting together diagnostics from multiple platforms at the same time, you can see what's happening with the other platforms. In addition, sometimes you need to put together groups of experts to set a reference standard and agree on what was going on with the patient, and also what those results might mean, whether they make sense in terms of what's happening or not. And so we've put together a paper that's under review on reference standards. But I think if you do a platform study like we talked about this, having sort of a centralized reference standard can really benefit all the diagnostics that are participating in that platform study to get closer to the truth. And quite frankly, maybe at some point to redefine the truth or redefine knowledge about what we know to actually be going on with Vance's patients. Because I think, like with pneumonia, what we do today is pretty poor in terms of trying to figure out what's going on in those patients' lungs. And having that normal microbiome, right, all the way from the mouth down into the respiratory tract is really a challenge for a lot of our diagnostics.
I actually have a question for Robin, and thank you. That was a beautiful answer. So have you used panels? So depending on your specimen type, which could be, the one you just discussed that's got a long track, or blood, there's always the potential for contamination, for example, skin flora or other contamination. Do you go, if you've got a master protocol that's got 5 companies or research groups, do you go with best 3 out of 5? Do you have an external panel who says, okay, looking at this sample, blood culture detected strep here, oralis, so something, maybe the patient got their teeth cleaned earlier that day or something. Do you have a panel that says, okay, the blood culture saw it, one of the other tests saw it, the others didn't, there's a good chance it was only in one of the culture bottles. How do you determine? There are many ways of determining truth, and it's not always a pathogen. So how do you approach that?
Right. I think all of what you said, but I think in the end, you likely need an expert panel who can review all the data and look at that and interpret it. You know, you gave that example. I can also give an example of a direct from blood diagnostic where blood cultures are negative and the patient has cellulitis and you detect strep pyogenes. Well, if you're using blood cultures as your reference standard, that's a false positive detection. But all of us would sort of say, well, wait a second, you know, that actually kind of makes sense. And it's probably true positive and the blood cultures negative. In some ways, I would argue that the way we use blood cultures is not really to diagnose bloodstream infections. It's to diagnose infections at other sites like pneumonia and skin and soft tissue infections. So some of the terminology we're using is just wrong. And some of these newer diagnostics may be better at detecting what's going on in sites outside of the blood. But to sort all of that out, I think we need experts to get together and try to get closer to what the truth might be. In the end, of course, we need action taken on behalf of those results and to show that that's the correct way forward for our clinicians like Van. So it is complicated, Melody, but those are my thoughts.
Excellent, thank you. I think for my last question, you know, I think a number of us are always trying to think through what can we learn from the last 2.5 years that would be applicable. And Fred, you gave some nice examples of what you saw kind of over the counter for COVID-19. How do we encourage that the type of perhaps innovation or regulatory streamlining that are specific to the type of products that we're discussing today?
Well, diagnostics have changed much for the better, I think, during the last couple of years. I think it's both on the clinician side, they have a better understanding of the role of diagnostics, as well as the public. You know, the public now, I think, is really into doing their at-home COVID test, and the whole movement of diagnostics out of the laboratory into point of care and into the home is something we have to pay a lot of attention to. Overall, I think it's a good thing with a couple caveats. Number one is public health. If people are testing at home and they don't report, then how do we know how widespread COVID is? Or as we move, you know, STI testing into the home, how do we keep track of who's got gonorrhea and how do we do our contact tracing? These are issues we really need to think about a lot as this diagnostics evolve out of the reference centers and into other places. Having those tests available in 15 or 20 minutes is great. It's very important, but we have to think it all the way through to make sure that when we're doing this, we're doing it in a smart way, both for patient management as well as for public health.
Yes, Robin?
I completely agree with Fred, and I will add a couple of other thoughts. I was serving as ASM president when we came into the pandemic, and so we were thinking about what a society needs to do faced with the pandemic, and we had many thoughts, and I'll just share honestly, one of my many thoughts is, we need to encourage diagnostics to get out there, and we need to encourage essentially reimbursement for all of the diagnostics. And we thought about it, and we did not advocate for that because we knew that would happen. We knew that this was the obvious thing, that there would be incentives to develop diagnostics, to get them approved, and also to pay for them. And I think that's something that we don't have in the area of antimicrobial resistance. And that's something that we can learn from what we just experienced with the COVID-19 pandemic.
All right, excellent. That's a great way for us to end this panel. I want to thank all of the speakers for your remarks. And with that, I'm going to turn it over to Mark for session number 2.
Thank you.
All right, thanks very much, Mary Ann. Thanks to all of our panelists for an excellent discussion of some of the challenges and really interesting developments with respect to innovative diagnostics. And Robin's last comment about reimbursement is a great transition into this session. During this second session, we're going to talk about the market for bacterial diagnostics, including general bacterial diagnostics, as well as those that identify drug-resistant infections and antibiotic susceptibilities. And I appreciate the previous panel making those distinctions, which are increasingly important. Specifically, we're going to talk about some of the current financial incentives, some of the uncertainty around that, as well as opportunities to facilitate and sustain development of and access to innovative diagnostics as a key part of this overall strategy on effective AMR management. Very pleased to have our panelists with us for this session. Mara Aspinall is Managing Director at Health Catalyst Groups and Group and Professor of the Practice at the College of Health Sciences at Arizona State University. Mara's a long time, thanks to COVID being around for a while, long time collaborator with Duke-Margolis, including on issues like that RAD-X approach that we heard about in the first panel that might be applicable here. Also joined by Mark Miller, Executive Vice President and Chief Medical Officer at bioMaru, and Fanon Solomon, who's Director of Market Access and Payment Policy for Becton Dickinson, and Betsy Wonderly Trainer, who's Alliance Director at CARB-X. So great set of panelists for opening comments. And Mara, I think we're going to start with you. And I believe you've got some of your own slides to project.
Yes.
Or do we have those? Okay.
Thank you. And wonderful to be here today. Can you all see the slides?
Yeah, you're not quite in presentation view though yet. There you go.
Okay. Great. A little bit of a delay there. So it is great to be here, and particularly in listening to all the great speakers in the first session. So I'm going to go through these quickly, but talk more about diagnostics than specific to here, but put together some operating models that might be useful. Although I have to start on a little bit of a light note, because as I was trying to look at the most recent data and publications on antimicrobial diagnostics, guess what came up? This, the mold that changed the world. And indeed, this is a new show in Edinburgh. And I'm told it's sold out. And this is not my comments below, but I love this. A musical about antibiotic resistance may not seem like the most entertaining of subjects, but indeed, it's informative with the best of musicals. And when they talk about it, they say infectiously catchy songs. So if we need to educate the public, just like Hamilton created a new interest in history, then maybe we need to do this through entertainment. I can't see your faces, but I hope that somebody is smiling as you're taking a look at that. So, but on a more serious note, just a few slides here. We've talked about it, but I thought this was a very powerful graphic that looks at the rise of resistance and the amount of time now measured in less than 2 years where we're finding antibiotic resistance for common drugs, where in years past we were getting 10, 20, 30 years, we're now getting 10, 20 months. And all of you know this, but I think it's very powerful when you see it all together. You also know that this is very much the case that many of these medications don't work broadly. So there's sort of a sense that it's okay for an antibiotic not to work because we have so many drugs from diabetes, arthritis, and Alzheimer's drugs, and clearly cancer, that don't work. And a comfort level, particularly amongst many physicians, that says it's okay if it doesn't work. Let's just, don't finish it and try another. It's been a lot of education, but still not enough. So why are diagnostics important? It's the core of this meeting. And I loved in the first panel, people say that they love diagnostics. So I'm going to provide a little bit of context here in diagnostics bridging theory to therapy over time. If you look at diabetes, it was only when we had the drug and the diagnostic that it became treatable. If you go to infectious disease broadly, microbial testing and antibiotics, and clearly that's what we're here to talk about. But let's look at liquid tumors. chemotherapy, Hodgkin's, this was all became much more treatable when we had the basic diagnostics. On solid tumors, the same is very much true in 1998, 1965, the very first biomarkers, and then the first companion diagnostics. But maybe most clear is on AIDS. It's only when we had the ability to do the tests could we fully use AZT and other antivirals and make the life expectancy for AIDS equivalent to anyone else. So it's the diagnostics working with the theory that makes the difference. So how has that evolved? I think about it as 5 stages. We start with, is there disease? Diagnostic 1.0, what disease, what subtype? Can we identify the risk, and can we preempt disease? But the key thing here is about the tools. We started with just MD5 senses. We moved to manual systems, automated lab systems, algorithm data analytics, and the future is, I think, predictive AI with the diagnostic, the therapeutic, and the data. And there was a lot of discussion in the first session about using that data both across company, across facilities, across patients, so we can truly predict what will happen and educate physicians and avoid resistance. But to do that, again, we talked about this, the core here is the medical model versus the population health model, or what I call individual versus public health. And on a day-to-day basis, it feels like we're focused on an individual patient. But in this way, we have to be focused on a population health model and disease prevention, not, I'm going to say, not just the individual. But for a diagnostic company, that becomes challenging because on a day-to-day basis, on a financial model, it's all about getting the clinician what they need on an individual basis. And to put this in context, what you can see here is that patient journey from screening to monitoring has all sorts of questions. But the real question is the so what test. A physician needs to feel that it's so what. When I ran a large diagnostic company, I was asked a lot about who are our competitors, and everybody thought the answer was Quest and LabCorp. And the answer was doctors' brains. So the key here to me, more than almost any other area, is how do we put the right incentives together and the clarity that doctors, and somebody said this, that doctors want to give the test, and that any of the diagnostic companies involved can answer that so what test, not just for the individual patient, but for the population. So in summary, for my initial remarks, if we could talk about it, I like acronyms, so we're going to call it SPEED. 5 areas. 1, speed up R&D to approval to market through collaboration. I think Robin said that. I happen to be on the board of the Critical Path Institute. There are virtually no work on pre-market approval collaborations in diagnostics. Almost all of them are in therapeutics. How do we change that? Number 2, process integration, integrate into current systems without disruption. I like to call it be disruptive without disrupting, because physicians are historically not happy with changing their processes. Education, I call it acute and chronic education. We get a lot of acute education when there's some crisis here. It needs to be chronic in terms of antimicrobial resistance. Efficacy, you all talked about this, but I'm going to be somewhat controversial here, maybe. Not enough labs, not enough companies are publishing, and not enough of the biggest journals are publishing diagnostic data. How do we work together as an industry to get more of it published and get it in the biggest publications out there? And lastly, incentives to develop products. We'll talk more about that. In particular, I call them companion therapeutics as opposed to companion diagnostics. How do we make those the standard? So with that, look forward to the discussion.
Mara, thanks very much for that overview. Really appreciate it. And next, I'd like to turn to Mark Miller at bioMérieux.
Great. Thank you very much. Thank you for the invitation to come and speak here. It's a great group of people discussing an obviously important topic. So the only, I have no slides, so already you should be cheering. But my caveat here is that what I'm going to be talking about in terms of market access really applies to a company like bioMérieux, which is an international company with reach into both developing countries and developed countries. And so the focus has to be quite broad. We have to consider a global customer base of labs, hospitals, patients in both of those areas, developed and developing, in 6 of the world's continents. So every time we develop a product, it can't just be for an American market, for instance. And a lot of the discussion here, although it's been fantastic, has been very, shall we say, Americanocentric. There are many developing countries who need basic diagnostics, where a lot of the AMR is located, and they need basic microbiology. And anytime we create something innovative, there's always a higher cost involved with that. I can't think of any innovative test that costs less than what we have today. So innovation usually equals more costs, and more costs are not really consistent with emerging markets. So we have to have both the developing world in hand in terms of providing good diagnostics for AMR, and we also have to have the developed or the high income countries in mind, where they want innovation, they want faster, more actionable diagnostics, especially for AMR, and we have to please all of that. The other caveat about being a larger company and my talk is that, what I'm saying may not apply to startup companies who are developing diagnostics, and, push and pull strategies for them are very different. One example is, prizes and some limited funding for developed diagnostics. For a startup company to give them $5 or $10 million in order to develop a new diagnostic usually is a bonus for them. It's great, and it convinces them to take on a project. For a company like ours, $5 to $10 million may obviously leverage and help with the diagnostic development, but it pales in comparison to the market access costs which follow. And so if we don't get assistance, and I'll get to that after, but if we don't get assistance for the market access, then what's the point of developing the diagnostic test, because we can't get it out there. So talk about getting it out there. It's much more difficult to deploy, and this is one of the reasons why diagnostics are difficult for everybody, and I've heard the pain in everybody's talk. Deployment of diagnostics is much more difficult than therapeutics. For therapeutics, and I don't want to minimize, getting therapeutics out there, but you need a good transport system, and you may need a cold chain. And although, sometimes it's very difficult, for diagnostics, we have usually diagnostic equipment and reagents. And so we need the equipment and reagents to get where they're going, but we also need training and education, equipment installation, storage facilities, a quality control program, equipment maintenance and service, software and hardware usually, connectivity, and some type of power supply. And so when you put all those together compared to, massive drug administration for parasitic diseases, where you give a combination of 6 pills in a cold chain controlled transport, diagnostics are much more difficult to deploy. In terms of market issues, AMR-focused diagnostics suffer the same problems as antibiotics. And I know there's been a lot of focus on the market conditions for antibiotics, but they really are no different. And, Robin touched on it, and Fred touched on it, and, it's exactly the same thing for us. For certain diagnostics, there isn't a big market, there aren't big margins, and we have to be incentivized to bring them to market. On top of that, we have much shallower pockets than a lot of our pharma brothers and sisters, and our margins are much lower. So we really do need incentives, almost more than they do, in order to bring a product to market. And siloed budgets are killer. So when we try to justify bringing diagnostics, especially for AMR, to market, and we justify the cost of the diagnostic by saving antibiotics, saving length of stay, decreasing hospital resources, decreasing morbidity, the argument always is, oh, that's another budget. You're saving money somewhere else. That's not in the lab, that's not in the hospital, that's in the community, that's outside. oh, decreasing AMR, that's a theoretical benefit. You know, we can't count that. We can't monetize that right now. So all you are is an additional cost. You're just an additional diagnostic cost. And the savings are somewhere else. And so that makes it very, very difficult. Diagnostics are complex and expensive to create, develop, produce, and get market access for. We have the same complex market, as I said, as pharmaceuticals. And we're constantly asked, and I'll give you an example, you know, why do you not develop a good diagnostic test for invasive fungal infections? You know, why can't you do something better for Aspergillus or for Mucorales? And the answer is we can. We and many other companies have developed diagnostics for invasive fungal infections, but the market is very small and there is no incentive to bring those to market. We had a very interesting discussion a couple of months ago with the CDC. There's a number of us from diagnostic companies who were on the call, and the CDC gave us all the epidemiology of invasive fungal infections in the US and said, why aren't you developing diagnostics? And a number of us said, we have diagnostics for these diseases. Is there somebody out there that we can partner with? And again, I love Fred's partnership talk. Is there somebody out there to partner with us and to help us and to leverage this so that we can bring them to market? You know, can we get a BARDA grant? Can we get the NIH to fund these? Can we get other leverage so that it'll make it interesting for us to bring this to the market? And the CDC's answer, as we hear a lot, is, oh, that's not our business. We don't do that. So as long as people don't get into this spirit of collaboration and partnerships, you know, it's always going to be the same question. Why aren't you developing a diagnostic for whatever it is? And we say, well, there's really no market for it, or we need some incentive to bring it to market. My last point is probably the most important. My last point has to do with a number of things that the speakers already said, which is value. Right now, diagnostics are not reimbursed or valued according to what they bring to the patient or to society. So, for instance, almost all diagnostics are reimbursed and valued based on the technology. So an immunoassay, regardless of what it does, is reimbursed as an immunoassay. My classic example is a vitamin D immunoassay. As good as it is and as useful as it is, it doesn't help us to combat AMR. A procalcitonin immunoassay is reimbursed exactly the same as a vitamin D immunoassay, even though procalcitonin can be a remarkable tool for antimicrobial stewardship. So the value of PCT as an antimicrobial stewardship tool is not valued in any way. Therefore, the reimbursement is not there, and therefore it's not used like it should, because it's reimbursed like a vitamin D immunoassay. We have to start valuating and evaluating in HTAs, health technology assessments, and reimbursing diagnostics for the value that they bring. And that value has to be harmonized, clear, and standardized across countries. And that's it. Thank you very much, and happy to participate in the questions.
Great, Mark. Thanks for presenting very eloquently a lot of challenges, maybe some directions forward. Next, I'd like to turn to Fenan for your perspective on some of these issues related to market access and payment incentives.
Thanks, Mark. Can you hear me?
Yep, just fine. Thank you.
Perfect. Thank you. So yeah, a lot of my colleagues have touched on some of these things already. Hate to be repetitive, but I guess I'll be hyper-focusing on just, I guess, one logistical aspect of the issue, but some great points have been made. So there are several factors that disincentivize the manufacturing of innovative clinical diagnostics. For one, the existing CPT codes that an innovative test is forced to use at launch are typically based on older tests that correlate with reimbursement rates that are so low, as Mark Miller just mentioned, that clinicians are actually deterred from utilizing and paying for those tests, especially if you think about smaller physician office labs that constantly have to keep their P&L in mind. Two, if you want to obtain a new CPT code that's more reflective of the test description and reimbursement value, it's quite difficult because the process isn't really that transparent. So for example, high test volume is just one of several criteria, but that number is not clearly defined anywhere. And just keep in mind that, you know, all of this happens before a manufacturer can even approach a payer with any clinical or cost benefit data. So if you're on the market without that differentiated reimbursement, you're potentially inviting the bulk of your physician or lab customers to participate in a multi-year saga of revenue loss, which nobody wants to do. And then if, by some miracle, you've gotten over that hurdle, then come the cuts eventually. So payment rates for diagnostic tests that are set by CMS are subject to cuts up to about 15% as laid out in PAMA. And it's based on a complicated equation for data collection periods that are years behind. I mean, there's such a huge lag. And then I just overall, I think everybody can appreciate, maintaining the integrity of the AMA CPT code set, but I think there has to be some transparency around the rules, and that's just not the case today, which is just really one of the logistical factors that serves as a hurdle and deters companies from wanting to get into the game in the first place. So I know it's such a minor aspect of some of the overarching themes that some of my colleagues brought up, but I wanted to make sure that that didn't get lost. Thank you.
And thanks very much for your comments. Definitely sounds like some challenges here. I'm going to come back to you all in the discussion part for more steps that we can do about it. But first, I'd like to turn to Betsy to talk about some of the policy steps that could support diagnostics in these efforts to combat antimicrobial resistance. Betsy?
Absolutely. Thanks, Mark. Let me just get my notes over here. So I too skipped the slides, so you can all celebrate on that. I do want to kind of come back to some of the comments that Mark and Fernand mentioned, but first we are, I think, on a good note, a result of a lot of the policy changes and the evolution that has been happening in the AMR space. CARB-X, for those of you that don't know, is a public-private partnership that was really driven by the realization there was need for change in this space. And so we are really providing targeted funding and programmatic support to accelerate the development of therapeutics, preventatives, and diagnostics that are addressing antimicrobial resistance. So we're funded by BARDA, the Wellcome Trust, the German government, Gates Foundation, GAMRIF, and we received in-kind support from the NIH. And we received about just under $500 million in our first 5 years, and we've just signed our new contract for the next 10 years for a similar amount. So we're excited about being able to provide some push incentive to support this space across 3 different pillars. But we also realize there are a lot of challenges after the life of CARB-X. So just a little bit about the diagnostics program within CARB-X. We follow a strategy that has been coined Align by Design, as we're focused on ensuring that the therapeutics and the preventatives in our portfolio have the appropriate diagnostics to support their clinical trials and market uptake once they make it to the market. As such, if you take a look at our portfolio, it's primarily diagnostic investments in technologies that are focused on bloodstream infections, lower respiratory tract infections, urinary tract infections, and sexually transmitted infections. And again, although we're primarily focused on research and development, and for diagnostics, we fund from technical feasibility through product development, we're keen to be supporting the programs that are in our portfolio so that they can be successful in the market when it comes time for them to launch. And so we spend a meaningful amount of time thinking about life after CARB-X with our companies and helping them prepare for the next chapter. Some of the issues that were mentioned earlier about accessing clinical samples and the expenses of these studies are a lot of examples of some of those challenges. But in addition to that, we have some graduate companies that we keep in touch with and we receive good feedback from in terms of some of the market and policy related challenges that they have faced or they are facing as they bring their new AMR focused diagnostics to the market. And I want to just mention a couple of those. In most of the companies that we've funded to date are smaller companies. They have a little bit of a different perspective from bMérieux and MBD and Cepheid, but there is some overlap. So I will mention overlap just so that we all realize it's a common issue. But I think some of these might be a little bit new to this discussion. So starting on a positive note, Fda Breakthrough Designation, I think is seen as a very helpful thing for a lot of the companies that are getting into this space. It helps raise awareness about the company's technology to potential strategic partners, investors, and future end users like Vance. We all like to refer to Vance. However, I think the availability of funding for the procurement of the AMR-focused technologies that they're developing is quite limited, as we've discussed. And because the antibiotics are so low, the price of the antibiotics is so low, the product uptake for these small companies that are really needing to generate revenue as they come to launch is often really slow. And so this brings me to the NTAP. I think the new technology add-on payment is helpful. So thank you. But there are some limitations. And I think one is the time limit to this. And I think it's 2 to 3 year time limit. And I think this is often, as Fanan mentioned, a multi-year saga of revenue loss, which I think is a great way to put it, very visual. And this has been really challenging for some of the companies that launched right before COVID or launched during COVID. And so I think, NTAP is an area where we are walking before we're running, but I think it's an area where we could possibly think about how we could evolve that program a little bit more. And importantly, as Mark mentioned, there is a disconnect between the Microbiology Lab budget, which is responsible for product procurement, and the hospital budget, which receives the reimbursement for products which are procured by the lab. And as such, the NTAP carries very little value to the product procurers in the micro lab because they struggle to recognize the reimbursement benefit that comes much later to a different budget. And so, many companies have shifted to a reagent rental model, as this disconnect really makes capital purchases, nearly impossible, or at least very challenging. Okay, I'm going to just skip over a couple of these. I'll jump to just echoing what Mark said. I think we spend a lot of time, diagnostic manufacturers spend a lot of time trying to demonstrate value through large clinical utility studies that have to happen at multiple different sites with different entities. And I think they also have to market their products to multiple silos in each of the hospitals. And I think this is very expensive and time consuming. And perhaps if this could be more focused and more efficient, if the financial models made more sense. So I'll pause there and just thank the Duke-Margolis team for raising awareness around the need for better financial models in the space in order for the diagnostics that we all value as meaningful tools to be used to support both near-term patient management and long-term public health issues. As we saw with Mara's slides, the growing resistance rates are continuing to grow. Thanks.
Great. Thanks very much, Betsy, and all four of you for laying out some of the challenges and opportunities. I have to say, after hearing from this panel, I think the challenges, heard a lot more about those than the opportunities. So I'm going to push in this, the last 15 minutes or so that we have together on the opportunity side. You all mentioned some steps that have been taken, for example, the new technology add-on payments that have helped at least incrementally with some therapeutics. We heard from Betsy about some of the support that CARB-X is providing upfront to help get closer to market. So both on the push side and the pull side, let me open up just first for any opportunities that you all would like to get on the table that we haven't talked about enough already. And thanks all four of you for coming back on video for this discussion. What are some promising either regulatory actions or, if needed, legislative actions that could create better incentives for the development and especially getting those further steps as we heard about from Mark, these further steps needed to get more of these products actually into the market? Anybody want to start?
I'll start again with a broad piece, which to me it's, and everyone's touched on this, which is the importance of the regulatory process. The VALID Act may be doing some of that. We didn't talk about that. There are pros and cons to that. But diagnostics have always been an orphan in some way as part of CEDAR. I believe that what VALID might do is create a separate regulatory system that's truly focused on diagnostics. And if we think about how diagnostics compare with therapeutics, therapeutics have been heavily regulated on approvals and lightly regulated on price. Diagnostics is the opposite. I should have shown this slide, where diagnostics are heavily regulated on price and lightly on approvals. And I would argue and emphasizes the points that everybody's made from a diagnostic company, the margins are so thin. So whether it's VALID or something else, we need to change the regulatory environment in order to change the reimbursement environment. So I'm somewhat optimistic, but I don't want to be naive.
Great, thank you. Other thoughts?
Well, I think there's been positive. Certainly there's been positive, and I didn't want to end on a negative note, but there's a lot to do. I think getting back to what was said at, CARB-X really has evolved. And I think, years ago, there was no funding of diagnostics. There was funding of small antibiotic companies. And when they got their molecule to a point where it could get to market, they had to find their own funds to develop diagnostic testing. And there was no funds available. And, none of us do this for free. So they wound up at a dead end. And I think that, CARB-X came up and there's been a lot more funding for them, these small companies with novel antibiotics or novel anti-infective approaches, whatever it might be, in order to fund the diagnostics as well. And that a real product has to pass that second valley of doom to get into the marketplace, and it requires a diagnostic to get there as well. The other thing I wanted to say is that there's been a lot of progress. If we look to our brothers, sisters, cousins in oncology, there's been a lot of use of diagnostics to develop oncology drugs, so-called companion drugs. We tried to do that a few years ago. The model doesn't quite work in infectious diseases because the anti-infective companies only look at this as reducing their market. But, I think that if we take a lesson from oncology, it's that even in oncology, which is a success for companion diagnostics, the diagnostic is still not valued for the value that it brings. And so I'll just give you one example of, for instance, BRAF mutations for melanoma. And I know that one best because we developed a test. And, a patient will benefit for melanoma treatment based on the BRAF result. That treatment will bring to the pharma company $40,000 to $45,000 a year for the lifetime of that patient, which can be 2.5 years. And the diagnostic company that provides the mutation knowledge to allow them to get that treatment gets a single payment of $285. So the value to that patient is not transmitted in the whole scheme. And I think that we see this repetitive non-valuation of diagnostics in AMR and in a lot of other medical spheres. Obviously, we're talking mostly about AMR today, but I think this valuation, what is the value of a diagnostic that saves you using an antibiotic over and over again? There's got to be some kind of technology assessment that puts a true value on it.
Yeah, it's a great point. I think there are some steps in that direction. Again, more on the therapeutic side in England, elsewhere, that try to take account of some of that population value, which, as you said, it's an important issue for antimicrobial resistance. It's frankly a more generally important issue for infectious diseases as a whole because of the impact on transmission and population impact of steps that reduce the forward progress of any infectious disease. This is one reason that we spend so much time on these efforts at Duke-Margolis. But I really appreciate the comments about getting to value. Any others that you'd like to add? Because then I'm going to go through my own checklist and see about getting some reactions based on what we've heard so far. Okay, we have about 10 minutes left. So what are some policy steps, feasible policy steps that we could help? Can we spend maybe a minute or two kicking the tires on each of these? Obviously, as Mara started out with, we need to start with a musical, something that's going to raise public awareness. I see from Nicholas's comment in the chat that I guess lucky for us, the new musical on the mold that changed the world is coming to Washington, D.C. So maybe we can get some of our policy friends out to see it. So maybe that's step number one. Step number two, we talked about some of these push supports that CARB-X is already providing to some extent. Have we checked that box? Is there more? I mean, I guess there's always more that could be done, but it seems like you all see the critical gaps as a little bit further downstream. But let me just confirm that point. Definitely some appreciation in this panel for the work that CARB-X is doing on helping to move diagnostic product ideas forward from the development concept into getting closer to market.
Maybe I'll start on that one. I think we do see a lot of funding pre-CARB-X. So the NIH provides a lot of SBIR grants, and there's a lot of concept phase and early development funding available, which most of the companies that come into CARB-X already, have accessed. And then when we stop funding at product development, it's really at the integration of the alpha prototype. So once the instrument, if it's a system, a larger system with instrument cartridge and software, once that all comes together and gets out into the hands of external users and get some data generated, that's really where we stop. And so I think there is a need for additional funding beyond that. BARDA, the Advanced Development Program, usually, starts to look at companies around then, and then the private sector. Some of our companies have received investment from private companies after that, but also strategics. The larger IVD companies, I think they're really looking at that stage to try to partner with someone who won't only fund the rest of the development, but will also be able to leverage their commercial footprint and all the work that they've already done in laying the groundwork in the AMR space. So I'll just kind of set that ground.
Right.
Yeah.
Yeah, that's some great points. I'd say too, AMR Action Funds express some interest in this general topic as well, and an idea of getting some private sector funding partnerships to move closer to market. Obviously,
they're only drugs. They don't do diagnostics.
Yes. Well, the other big topic that came up was better evidence development, and maybe divide that into evidence before a treatment comes to market to help to get to approval and to help to get to an initial covers decision. You all talked about the independent test assessment program. We heard today about ITAP and the RADx efforts around COVID and OTC tests. Let me do a reality check on whether you all think something like that. If there was public funding for a framework to do validation testing to get to more confidence about sensitivity, specificity, predictive value of tests in this context, is that a way to bring down costs and increase progress to market significantly? I see head nodding, yes. And any different issues here, or that's a good model that could potentially be applied to some of the new innovative.
Well, I was impressed by the RADx model, I have to say, and I see from some of the speakers it's
not that expensive either, in the scheme of
no,
things.
it's not, and you know what it did? It took diagnostic tests, which a priori didn't have what people wanted in terms of sensitivity and specificity, but said, hey, if we put them into particular contexts, whether it's daily at school or at home or whatever, does it actually work and deliver the information of value that we need this to deliver? And they showed that, regardless of the in vitro sensitivity and specificity, that in real life, it delivered what it had to do, in certain situations. So, I like that. That's what we all try to strive for, that true health economic value, the real world evidence, when you put something into the real world, and does it suit the objective, getting back to what all the previous speakers at the previous roundtable, does it answer the particular clinical question that helps all of us, especially the patient? So I thought it was a good model. I'd love to see it for non-COVID tests. Yeah.
Sounds like an area for further development of what would that look like in this context. Next, Mara brought up potentially some opportunities for regulatory clarity and streamlining, especially in the context of VALID, which would provide, as Mara said, a clear framework for diagnostic tests. That has been controversial, with many of the organizations that have supported and developed so-called lab-developed tests, locally developed tests, about worrying that this might disrupt innovation. It does seem like if that is going to come together, VALID is going to come together this year, it's going to need a path forward, maybe some modifications that highlight areas like this one, where more clarity really would be helpful and would be a good basis for perhaps an FDA set of initiatives related to clarifying regulatory pathways for getting to confident tests. So I just put that one out there too. In the last few minutes, I want to turn to the coverage and payment side. I think, Betsy, it was you that talked about some of the work or some of the benefits that a breakthrough designation can have. That's something that, again, could be further developed on the FDA side, but also has some implications with CMS, where CMS has tried to take some steps and announced they're going to be doing more in the area of breakthrough devices, so that companies can come to CMS early and get helped with questions about what remaining evidence gaps are there from the standpoint of CMS coverage. Remember, there needs to be a coverage category. It needs to be reasonable and necessary, not safe and effective. It's not exactly the same thing, and more about relevance to the Medicare population of the safe and effective, in this case, tests. And also maybe some help with those CPT pathways that Fanon talked about a little bit as well. Is that an area that could be helpful, sort of a CMS side to the breakthrough pathway, like we're expecting to see from CMS in the coming weeks and months on breakthrough devices? No, any thoughts?
I feel like I'm the least.
We're at the end of time, but it's got to be quick. But
I mean, I feel like I'm the least well-suited one to comment on this. But I think it's the disconnect between the budgets that's a bigger issue. I think it's the lack of the micro lab who's procuring the product, the instrument, the cartridges. They never get that reimbursement back. So. So that's, I think, where the primary focus should be. I know I'm answering your question directly, but I also realize there's 3 minutes.
Yeah.
So I just want
I agree.
Sorry. I'll just add to that to say, from a diagnostics point of view, we have to focus on having our providers and payers look at full system costs. Not just the diagnostic, because we will always lose if it's just the diagnostic, but it needs to be system cost.
And that's exactly where it's going next. And sort of the final step is getting to payment that more reflects value, which, as you all pointed out, there's not really a mechanism for that now, and that may be most important. This is an area where there has been a lot of recognition on the AMR Therapeutics side, with bipartisan, actually fairly broadly supported, Pasteur legislation. It seems like even if Pasteur just applies to therapeutics, if done well, it could actually help with the diagnostic side. I think it was Mark, you commented that pairing a diagnostic with a therapeutic may not be that valuable now if the therapeutic is being paid based on on volume. If it's paid based on value, where over time your Pasteur payments are linked to demonstrating that you're actually preventing resistance from spreading and having a population impact, that could be a change. And I don't know that there's been much appreciation for the diagnostic therapeutic interactions in that space. But more generally, if you all have any final comments in our last 30 seconds on ways to have metrics for value of diagnostics that could be part of payment, that seems like coming out of this discussion, a really critical gap and potentially an opportunity.
Yeah, I just want to, oh, go ahead, Mark.
No, and I'm just going to say in 10 seconds, there are metrics. They're just not being used. I mean, we know the metrics that can be used in order to measure the value of a diagnostic. They're just not being used in practice. You know, NICE uses them sometimes. You know, Robin can put it together in probably 30 seconds because she does this all the time. I mean, we all know the metrics that will measure the true value of a diagnostic in combating AMR.
Great, thanks. And Fanon, you're going to get the last word.
Yeah, no, I was just going to say, you know, a test to treat methodology that is in place, meaning like market access is not just about payment, it's also about actual physical access to things. So having things in pharmacies, for example, having that software be, you know, seamless from start to finish, that's the best way to track things and make sure that you can get it to the highest volume possible so that people aren't deterred from going to the physician because it takes all day and things like that. So I think there are lots of opportunities that we're not taking advantage of right now just because it's not advantageous to. So
that actual delivery capacity that we heard about earlier, getting this into standard practices. But again, seems like something where metrics may be possible and could be helpful if linked to reimbursement. Well, you all have given us a lot to think about and a lot of opportunities for further work. Let me just conclude by thanking all of you quickly since we're already at 4 o'clock. We really appreciate your participation in this collaboration to combat drug-resistant infections and the critical role that diagnostics can play in that. As you just heard from my final comment, there is going to be some further Duke-Margolis work in this space, hopefully along the lines that we've discussed today. So this is definitely not a problem that's been solved, but I really appreciate everyone both bringing the big challenges here, but also concluding with some potentially very important steps forward for making progress. Thank you all for joining us today, and thanks especially for your commitment to working on this very important global public health problem.